Okay guys, we are continuing our journey through the different classifications of antibiotics here. We have already looked at cell wall antibiotics, and just to remind everyone what we want to remember for this particular exam when it comes to antibiotics and these classifications, we want to know all 5 of the classifications. For example, cell wall we just did, and we're going to do the group on protein synthesis now. We want to know the subgroups within each classification, and we would want to know an example of one antibiotic in each subgroup, okay? And it's going to get easier as we go. There were 5 subgroups within the first cell wall classification. This protein synthesis group has 4, okay? So it's going to get lower and lower. There won't be 5 in every category. But the names of the different groups within protein synthesis are chloramphenicol, aminoglycosides, tetracyclines, and macrolides. Now, we will find that with some of these subgroups, the actual name of the subgroup is the name of an antibiotic, so you don't actually have to know all sorts of other different names for every subgroup. In this particular case, with a protein synthesis inhibitor, think about how maybe you might be put on a cell wall antibiotic, and maybe it's not effective, let's say. Let's say that the bacterium causing the infection has no cell wall, like walking pneumonia, which we talked about a little while ago. Or let's say that the organism has some protective mechanism, genetically it's evolved to be resistant to the cell wall antibiotics. Well, then you might need to shift to a different category. And in this case, protein synthesis is the target. Now, you might be saying, well, this is not selective toxicity. This is not meeting one of the ideal characteristics of an antibiotic. It's not a magic bullet because protein synthesis occurs in our own cells. And you'd be, of course, correct that that is going on in our own cells. However, remember the ribosomes are different, right? So what we do is we use drugs like these that will key in on the reading of mRNA for the translation step. And in particular, it affects the 70S ribosomes that we find in these bacteria. All right, so here we have an image from a previous text we use, and they go through this in your textbook as well. How it is that there are 4 major groups, and here they actually name specific antibiotics. And you'll notice that chloramphenicol and tetracycline are in fact the name of the subgroup and the name of an antibiotic. Down here we have erythromycin and streptomycin, and we're going to go through these different subgroups. But I just wanted to show you this nice simple image, and they actually show you, which is what I liked about this one, is they show you an example and they tell you specifically what the antibiotic, what that subgroup is known for doing to interrupt the translation process. Here you have your mRNA, right? Here you have a tRNA. These are the transfer molecules that bring in the amino acids one by one. And here we have our ribosome, and that is, of course, our protein assembly factory. So let's go through each one of these in turn and talk about a little bit about them. Now, I'm going to give you some details about them, but this is just for your information, okay? It's not like you need to memorize every single detail about some of the interesting facts that I'm going to share with you here, okay? So chloramphenicol is a subgroup, need to know that, and in fact that's one of the drugs in the category. A lot of chlorampphenicols are broad spectrum, so they affect a lot of different kinds of bacteria, and they prevent the new peptide bond from forming. Okay, for this particular group of protein synthesis inhibitors, I would want you to know that is unique to the chloramphenicol group. Okay, so what do the chlorampphenicols specifically do? They stop this peptide bond from forming, so you can't actually have your primary structure of your protein. And a lot of these are bacteriostatic, which means they're just stalling the community, that particular population, I should say, from growing larger. Gives your immune system a chance. A couple other things I'll share with you about chloramphenicol. Chloramphenicol tends to be somewhat toxic, and so it's not a great alternative for bringing into the system. So it doesn't sound like it makes it a very good viable alternative as an antibiotic. But it is very easy to isolate. So when it comes to ideal characteristics, well, it's easy to get. It's cheap to actually produce. But it does have some overlap in its effect on protein synthesis, just a little bit, on protein synthesis when it comes to the host. All right, so we want to be careful in how we apply chloramphenicol. Being toxic, it's not such a great alternative for bringing into the bloodstream. However, they have used chloramphenicol to treat chlamydia in newborns. So if a pregnant mother gives birth to a child and the mother has one of this sexually transmitted infection, well, chlamydia is actually the top most preventable blindness that there is, because it's contracted to the child during the vaginal birth. So they'll use ointments. Sometimes they will use ointments with chloramphenicol to actually kill any bacteria that happen to have gotten into the eye there. All right, so what about aminoglycosides? This is our second group. Here are some examples. Some of the mycins, right? Streptomycin, kanamycin, gentamicin. All right. A lot of times, sometimes it's the prefix that gives you kind of the hint, like the cephs and the kephs back there when we were doing cephalosporin back in the cell wall antibiotics. Here we have the mycins cropping up. These will alter the ribosomal structure so that there's an improper reading of the mRNA, okay? And this is going to get in the way of the translation process. This is rather broad spectrum and bactericidal, so it usually kills. Now, a few details about this. In what respect is it broad spectrum? These are good against gram-negative and gram-positive organisms. They're particularly good against E. coli, Salmonella. They've been used against these in the past. Shigella, Pseudomonas. For Pseudomonas, it forms biofilms in the respiratory passageways. It's a particular problem for patients with cystic fibrosis. What they have done with some of these aminoglycosides, like for example, tobramycin is another example in this group. And tobramycin has been put into aerosols. So, almost like those inhalers that you might use for asthma or something like that. These are in aerosols, and you inhale them, and they can be somewhat effective against those Pseudomonas biofilms in the respiratory passageways. A lot of the aminoglycosides have fewer allergies than the chloramphenicol group that we just looked at. All right, so those are just a few interesting details. All right, our third group, tetracyclines. In fact, there is a tetracycline member of this subgroup, right? So the subgroup name has that as an example within the group. But if you notice again, we have this suffix cycling, right? So pretty much when you see that suffix, whether you see tetra in front or not, it is a member of this tetracycline group. And the tetra part, we'll see this in a second, tells you a little bit about how many rings happen to be in the molecules that are within this group as well. I'll show you some pictures in a second here. All right, so what are some details about tetracycline? Rather broad spectrum. These prevent the binding of the tRNA, so it's hard to bring in each subsequent amino acid. So you can't make your proteins. That translation process is going to be coming to a halt, and this is bacteriostatic. All right, so this is going to slow population of that organism from growing. Few details about this one. Broad spectrum, it's good against gram-negative, gram-positive. Aerobic and anaerobic organisms are going to be affected by this drug. Tetracycline was one of the earliest examples, and that's why it gets the name, this whole subgroup. But a lot of the later drugs, semi-synthetic, synthetic forms of the same basic type of chemical, were better in certain respects. One of the respects it was better is that it was retained in the body longer, that it has a longer life inside the host, so that it's able to kill for longer when it comes to the bacteria causing an infection. These members of this group have been used to treat UTIs, urinary tract infections, walking pneumonia. Now that makes sense, right? Because it's going to affect the protein synthesis of bacteria in the pneumonia infection someone has. And with walking pneumonia, the mycoplasmal pneumonia, any cell wall antibiotic wouldn't work, right? It's been used to treat lots of different STIs, so chlamydia, syphilis, gonorrhea, really pretty common. Unfortunately, as years go on and we use these drugs, a lot of those same sexually transmitted diseases are not treatable with these tetracyclines any longer because the resistance has grown among those populations of bacteria. So here are just some pictures. I don't expect you to memorize everything about these, but you see 4, right? Tetra, and they're cycles, right? They're these rings, basically, so tetracycline. Makes sense, all right? Okay, our fourth and final group within the protein synthesis category is macrolides. Now, this one, if you look at a picture, and I know sometimes people are like, scared of these chemical symbols and things, but a lot of times the pictures will tell you a whole lot. You see this big ring here, right? This big ring is the macrolide ring. Okay, this is why it gets this name, is it has this macrolide structure in the molecule. Now here we have our erythromycin as a common well-known example within this group. This binds and stops the movement along the mRNA, so the ribosome can't move. If it can't ratchet along to those codons, right, each 3 of those nucleotides in the RNA are going to code for amino acids, then it can't read the code, right? So if it can't move, can't make a protein. These are rather broad spectrum and bacteriostatic typically. These are mostly more effective against gram-positives. I'm not sure why, but they're a little bit narrower spectrum than some of the other protein synthesis drugs. And it all depends on what you're going for, right? If you want a narrow spectrum so that you can really narrow in on a particular type of infection, then you won't affect the normal flora in the body as much, and that's always a good thing. All right, so I think that's enough, guys, for this second category. Next time we'll catch up with the third, fourth, and fifth category of antibiotic type.